All right, continuing on with our talk about biotechnology, we had left off with a discussion about gel electrophoresis and how that works, about how essentially we cut fragments of DNA, drop the whole mess of fragments because we can't just say this fragment here and this because they're all too small and too tiny and we can't sort them out. So we're going to run through this porous gel to sort them out. And we do that by using electricity. The positive charge of the electric field pulls the negatively charged DNA through the gel, and the little pieces go fastest through all the pores, and the big pieces get held behind. And as a result, we get different banding patterns for different samples of DNA. We had already seen this slide. Now, why does this work? Why do different people's DNA, why does different, I can't speak, why do different DNA samples from different individuals have different banding patterns when they are analyzed this way? Well, of course, you might have guessed that it's because each person's DNA is a little bit different, okay? Maybe my pattern in a certain region is A, T, T, A, T, T, G, and somebody else's is A, T, T, A, T, T, A, T, T, A, T, T, G. Okay? And in this little region, I don't have as many A's and T's as they do in the same region of DNA. The thing you need to keep in mind about DNA, and we know we're all different, we know we're all unique and special and all that, but in reality, remember that genes code for proteins, and we want our proteins to work. And by golly, if somebody has a particular genetic sequence for hemoglobin, the protein that carries oxygen in your blood, and that's the telling how to build the hemoglobin molecule. Well, I don't want my genetic instructions for how to build hemoglobin to be different from anybody else's. I want my hemoglobin to work too. I want mine to be just like everybody else's that has working hemoglobin. So there are many genes in each of us that would actually be very much the same because we don't want them altered. In fact, natural selection has seen to it that they are consistently in one particular pattern to produce a particular protein. But there are places in the DNA where this absolute consistency is not so important because the region of DNA is not coding for something maybe as important as hemoglobin, or maybe it's not coding for anything at all. Okay? There are regions in the DNA that we call junk regions. Now, it may be that we've talked about the junk DNA, quote unquote, before when we talked about transcription and translation. In fact, it is slowly being discovered that these regions may not be junk at all, it's just that we don't know what they do. But there are certain sections that truly do seem to maybe not do anything. They certainly don't code for protein as far as we can tell. And there are certain regions of junk DNA that are actually made up of just repeated patterns, kind of like what I was showing up here. Okay? Here are some of the actual patterns you'll see. Sometimes you'll see cat repeated over and over, or GCC repeated over and over, and there are some others as well. Now, in a given region of the DNA, each person may have a different number of repeats. And there's many, many sites in our 23 pairs of chromosomes. And each of these may have different repeat patterns, and they may all, so let's get this picture straight. There's many different sites in all of those chromosome pairs where you're going to see repeating patterns, the CAT or the GCC or whatever the repeating pattern may be. And each of those, for each individual, may be a different number of repeats, adding up to a unique combination for each individual of these repeats. Okay, so let's look at this. Maybe one person has this number of repeats. Let's say it's GTA, or oh, there's the CAT, duh. All right, so CAT, 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 right there. So there's 3 here. This person has 1, 2, 3, 4, 5, 6 cats repeating in a row. This is the functional part of the gene we need out here. This is the functional part that may code for protein. This part is not coding for anything. It's just this region of this repeating nucleotide sequence. But because this individual's repeating sequence is much longer than this individual's, we would be able, if we analyze this DNA, to tell that this DNA did not belong to, was not 2 samples belonging to the same individual, but was actually 2 different samples from 2 different individuals. Okay, so let's see how we analyze this when we run it through a gel electrophoresis. Okay, so let's say our cut sites are shown as they are here. We're going to make our little sticky ends. This isn't in perfect alignment, but you get the idea. We'd make sticky ends here. Okay, so we're going to cut the DNA with those ever useful restriction enzymes. Okay, and when we do cut the DNA with our restriction enzymes, restriction endonucleases, we're going to get 3 pieces. This piece, this piece, and this piece. And if we counted up the nucleotides, 1, 2 pairs, the base pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 base pairs in this fragment. I don't know, let's say there's, I don't know, I don't want to count them all, 17, and maybe 13 in that fragment. So if that is true in those 3 fragments, then when we run them, we're going to dump all of them together in this well, because we depend on the gel to separate them. We can't do that, so we dump them all together in the well. And then what will happen is the yellow piece, that might be the longest piece, will go to this distance in the gel over a given amount of time. This piece, again, remember DNA is negatively charged, so it's being drawn toward the positive pole of the gel. I guess that would be this piece would travel the next furthest distance, and then we'd have one more piece, our shortest piece here, which would go even further. Okay, now if we had a second sample of DNA that had a much longer section here in the middle than this one, then maybe what we get is these 2 end pieces that are actually coding for something would travel the same distance, okay? But this piece, if it were much longer, would not travel as far as this one, and thus we would see that individual 2 would have a different banding pattern for their DNA taken from the same spot as individual 1, okay? So we can use this technique for all kinds of things. One thing we can use it for is diagnostic testing, or genetic testing, if you will, to see if someone is carrying a gene or that causes a genetic disorder. Okay, what we would want to do is compare a normal allele or gene variation. That's just a word for a variant of a gene, so gene variation. Like, for example, maybe you've got brown eyes and blue eyes. Okay, this is the allele for brown eye color, this is the allele for blue eye color. They both are, represent gene for eye color. They're just different types. Okay, so let's say that we know that allele 1 over here in this piece of DNA makes this banding pattern when it's cut with a particular restriction enzyme. We end up with 3 bands that are, this is the shortest band, it travels the furthest, this is the next shortest, and this is the longest band, it travels the least. But allele 2 is the disease-causing allele, and what we see is when it's cut with the very same restriction enzyme, it comes up with a very, very different banding pattern. We get 1, 2, 3, 4, 5 bands, and they go obviously different places. So that would tell us that this allele, even though it's in the same position as the normal allele, is very, very different. This would be an example of how a genetic test for something like Huntington's disease would be done. Huntington's disease, a deadly, ultimately genetic disease that causes people to basically lose control of their muscles and things slowly, gradually, until they are end up basically unable to function, unable to breathe, and it kills them. Unfortunately, symptoms don't show up until probably 30s, 40s, when most people have already had children and perhaps passed this allele on to their offspring. So just an example of how genetic testing could be used to see if someone has this allele or not. Okay? Forensics, crime scene type stuff. That's what we mean by this phrase in this context. Perhaps we would compare a DNA sample obtained from a crime scene with a suspect that we think could be involved in the crime and a victim that the crime was perpetrated on. So what we might have is a sample of DNA taken from a crime scene, and we can see that it's got 5 bands in particular order. And we look at the victim's DNA, and so we can see that the victim's DNA and the crime scene DNA don't match exactly. And then we look at 3 suspects. Do either of these suspects' DNA match what is found at the crime scene? And if you look at it carefully, you notice that suspect 2 probably has a DNA exact match with what has been found at the crime scene, thus possibly placing that suspect at that crime scene. Okay, so that's another way we can use this technology of gel electrophoresis. DNA fingerprinting, which is essentially what we just saw in the preceding slide. You're seeing a real example of DNA fingerprinting in this photo here. What we can do, various ways we can use it, comparing DNA banding between different individuals, stains on clothes, crime scenes, whatever. I think in the next slide, all right. So what we're looking at here is this is really what we just looked at a few slides ago, and I've kind of already done this with you, so I apologize for repeating, but what we're seeing here, again, we're going to cut this into 1, 2, 3 segments. This is one individual's DNA. This is another individual's DNA, but notice they have more of these junk DNA repeats. So again, 1, 2, 3. The DNA segments are going to look really different when they are strained through the gel. Okay, this yellow band is not going to go as far because it's much bigger. Yet these two would go the same distance, and we already did that. So my apologies. I didn't realize that it was coming. All right. I want you to at least have heard of this, RFLPs. Essentially, that is doing what we've just been doing. Here is what it stands for: restriction fragment. So these are the fragments of DNA that are made when we cut it with restriction enzymes. So that's what we mean with restriction fragment. These would each be 3 restriction fragments. Restriction fragment length polymorphism. In other words, maybe this fragment is that junk DNA, and in this person it's this long because they have a certain number of those junk DNA repeats. But in another person, maybe the restriction fragment is that long, much longer. Maybe they have more repeats. And so this fragment has many forms. It's a polymorphic set of repeats. There are many forms. It can have 2 or 3, or it can have 20 repeats over here and be much longer. And what we are doing when we analyze these junk DNA pieces, we're not really trying to get information about a particular gene. We're just trying to use these repeating segments that don't really code for anything to help identify, whose DNA is this, kind of a thing. And so that's essentially what we've been talking about. Okay, so it helps us to determine differences in DNA between individuals so that we can identify, was this individual at this crime scene or whatever. Okay. All right. Trying to decide if we want to go through this or not. I think this is really just rehashing what we've been over. Restriction enzymes would cut at particular sites, here, here, and here, for example. Okay. And so what we would get essentially would be 2 bands, this band and this one. Okay, and so this little one would travel the furthest down the gel, and this one would not travel quite as far. Over here, if we change the DNA sequence a little bit, okay, what we end up getting perhaps is only one slice through right here. Maybe there's only one restriction site because we've altered the DNA sequence a little bit. And instead, what we get is just a single band, because where it cut here before, it's not cutting anymore. And so we cut here and here, and we only get this one band. So what's going to happen is this is a much longer piece than either of these, and so what we see in this individual is that the fragment doesn't go very far at all. And then sometimes we even get weird things like sequences repeating themselves in genes. So what happens is we still get the same 2 bands, but this one is much longer than this original was at first because it's got this weird repeat in it. So what you would get is this one would go the same distance as that, but this one would not go near as far as this one did up here. Okay? Again, this is how these polymorphisms can be used, seen as differences in individuals, and used to identify DNA from different individuals. This is a real case being shown in this image here, showing the rape victim. Essentially what you have here are banding patterns, and I know it looks like big smears, but from the semen sample obtained from the rapist is showing heavy banding right here and here in the gel. Here is a blood sample. This is so this was taken from the crime scene. This is the suspect DNA sample, and what you see once again is heavy banding right there, okay? And that looks like a match, all right? And again, here's the rapist DNA right here, okay, for another sample, and again we've got another match. So we've got 2 samples showing a match both times. This could probably be used very strongly to convict a person of a crime. Okay? All right. Now, this is a murder trial. This is DNA coming from a murder trial. And what we see is we've got a blood sample from the crime scene, sample 1, sample 2, sample 3. Okay? And I just lost my pen. All right. And it's hard to see. It's very, very faint on this slide, but there's some bands that are being highlighted there. There's some bands that are being highlighted there, and there's bands being highlighted there. Okay, now what we have here is a blood sample from one victim, a blood sample from a second victim, and a blood sample from the suspect. Okay, so what we see in this image is we've got a clear match right here and right here and right here. Two samples from the crime scene matching a suspect, and we also have a blood sample from the crime scene that is tied to one of the victims. Okay? Now, what you should note, none of you were around, I don't think, when this was happening, but O.J. Simpson, famous football player, charged with murdering his wife and a young man that was accompanying her. Clearly, this is indicating a match between his blood and blood found at the scene of the crime. Nonetheless, he was acquitted of that crime, and that's showing the DNA of his ex-wife, or dead wife now, and the young man that was with her at the time. Okay, paternity testing, we can use DNA testing for that. What we see here is the DNA of a mother, the DNA of the child, and what we're looking for is this DNA from the father, or which sample is from the father. Okay, and what we would look at is to try to find, again, matches here. So let's look. Here is a band in this child that has got a match from the mother, but this band here does not. Mom couldn't have contributed that, but look, this guy could have. Kid has a band here, mom couldn't have contributed that, but dad could have. If it's this dad does not have it either. This dad does not have a match here. So again, what we're looking for is to see who could have contributed. Now, either of these guys could have contributed that stretch of DNA. Notice the kid does not have anything here, but this guy does, but the mom does not, so that could come from the mom. This here could come from only this dad. This one's a little off. Mom doesn't have it. This one came from the mom. This one here came from the dad. This guy couldn't have contributed that. So we would conclude from this test that this guy has to be the father. Okay? We can also use this technique to determine evolutionary relationships. Okay, what we see here are 5 samples of DNA from different species of organisms, and we are looking at each species on an evolutionary tree over here, and we can see that if we were to analyze this DNA, we could use it to see who was related to whom. I don't want to take the time to mess with that, but that's how DNA is used very concretely to determine relationships among different species of organisms. Okay? Now, sometimes when we are working with this crime scene DNA and things like that, the sample you have to work with is really small and you need to make more. As we said at the very beginning of all this, lots of times bacteria were the workhorses that would make more DNA. You insert the DNA you want into the bacteria, and the bacteria make more and more and more of it as they reproduce. Okay? But there has been developed a much, much neater, cleaner, more precise way to do that. And the method is called PCR. And you do not have to use bacteria at all. You just use DNA and some enzymes. This is called polymerase chain reaction, referring to DNA polymerase, which makes copies of DNA, which is exactly what we want to do. We have a small sample. We want to make it bigger. We want to make exact copies of the DNA we have. Okay? So this is simply a method that is very clean, very precise, and very quick for making many, many copies of a specific segment of DNA that we might want to test. You can do this using an initial amount that is incredibly tiny. 1 cell's worth of DNA is all you need. And if you can do this process on that 1 cell's worth of DNA, you can make all the DNA you need to run your tests. Okay? So basically, what you're doing in PCR is you're not requiring a living organism to copy the DNA for you. You're doing it inside of a test tube. All you need is the template strand of DNA, the original strand you want, you're copying from, a DNA polymerase enzyme, some nucleotides so that the DNA polymerase can add nucleotides as it's replicating the strands, okay, and a primer, which we know DNA polymerase can't do anything until a primer has been laid down. So the primers are critical, and you need to know a bit of the sequence that you want in order to make proper primers to put in. But these primers, if you know them, they can actually bracket the sequence of DNA that you want on either side, so you get precisely that sequence and no other part of the DNA. So you may start with a really long piece of DNA, but you just want a little short part of it, maybe a particular gene. Okay, so the primers are added that are of a specific sequence, and they kind of bracket the part of the DNA that you actually want to copy. And then basically what you do, put your DNA polymerase, the enzyme, the DNA polymerase enzyme, the primer, and some nucleotides together in a tube, and then you heat the tube. And the reason you do this is so that the DNA will denature, meaning we're going to break those hydrogen bonds going down the middle of the molecule. Okay? Now, once you've done that, DNA polymerase is going to go in, do its work, and then you're going to cool the DNA so that these pieces will come back together again, but you will have created more pieces. Okay? Trouble is this 90 degrees that you have to heat your DNA sample to. If you have to heat the DNA to that extreme, DNA polymerase, the enzyme, is going to tend to denature. So what you really need is a DNA polymerase that can withstand heat. And basically, they found it. There is a particular version of DNA polymerase that is called Taq polymerase, and it is found only in bacteria that grow in crazy places like these hot springs and whatnot. And natural selection has selected that stuff to withstand very high temperatures. So in using this particular type of DNA polymerase, scientists have been able to do this PCR reaction where they can just basically heat the DNA, copy it, cool it down, heat it again, copy it, cool it down, heat it again, copy it, cool it down, and the enzyme doesn't ever get denatured. It can handle that repeated heating. Okay, so that's it for this part of the PowerPoint. I think there's one more brief part we need to do, and then we'll be done with biotech. All right, so I know you're bored witless because I sure am, and I'll see you at the next podcast. Take care.